who here wants to know why they or their family member got their specific disease? Why did this happen? It'd be nice, right? So in the next hour, Brian's going to tell you why. Brian and I together are going to tell you why. We're going to answer all your questions. We've figured it out. We have, no, not quite. We're going to give you some best guesses. And we are really, truly honored to have Brian Weinshenker with us today. He's a professor of neurology at the Mayo Clinic. That doesn't come close to describing all of the appropriate accolades that apply to him. As I said at a meeting a while back, Brian's been doing neuromyelitis optica before anybody was paying attention to neuromyelitis optica. His research deals both on the clinical side and the basic science side. And when talking about autoimmunity for all these diseases, Brian and I share some interests, and he's been doing a lot of work on the genetics. And we thought when answering the question why, we'd split this into two talks with Brian tackling the genetics of autoimmunity, what sets up a host, and then I'll tackle the epidemiology of autoimmunity, the other side. And so I'm looking forward to the talk, and I'll turn it over to Brian now.
Thank you very much, Ben. And I'd like to thank Ben Greenberg and Elliot Froman for putting together this excellent meeting and for inviting me to speak. It's always a pleasure to connect with patients, and I've seen a number of patients whom I know personally, some whom I've seen at this meeting and other meetings. Also very pleased to touch base again with some key people that are involved with transverse myelitis and neuromyelitis optica especially. I'll just mention a few by name. Sandy Siegel, who he and I have now known each other for a number of years, and I really appreciate all the emotional help and support of the Transverse Myelitis Association for our work. And Victoria Jackson, I don't believe is here, but I'll acknowledge Victoria Jackson and Bill Guthey, as well as their entire staff, many of whom are here, of the Guthey Jackson Charitable Foundation and Greater Good Foundation, and all the excellent work they're doing on NMO. So today we're going to be talking about genetics of autoimmunity. And let me start. I always begin with this acknowledgement. This is really not going to be a major aspect of my talk, but I do have some intellectual property associated with the antibody test that's used for the diagnosis of neuromyelitis optica. And I think Ben has very well framed the next two talks. That's exactly how I thought I would start. The first question that I often hear after we give patients the diagnosis of an autoimmune disorder, whether it's MS or neuromyelitis optica or transverse myelitis, is how did I get this disease? And one of the corollaries of that is this something that has just happened to me? We may not understand exactly what happened, but it's something that happened to me, or is there something intrinsic in me, the way I was born, the way my proteins exist, that has caused this disease? And in particular, does this have some implication to some of my relatives? And for those that are parents, perhaps an even more pointed and poignant question is, can I transmit this disease to my children? And of course, part of that is there any way you can test for that? Can you test for certain proteins, genes, in a way that would predict whether my relatives' children are going to develop this disease? And I'm sure many of you have thought of these questions when you faced the diagnosis that you received. Now, of course, as scientists, your questions are really our questions, but we sometimes frame them a little bit more differently. So I'm going to pose a few questions, which is how a scientist would sequence them, and then I will try to address each of them and hopefully give you some answers to the questions that I've indicated that you have. The first question is there some indication of genetic influence? And as you'll hear, and in fact, Sandy Siegel, in a conversation that we had out in the hall, he told me, you know, I hear from a lot of patients with transverse myelitis, and they don't tell me about affected relatives. So why would we even think that some of these diseases have any genetic influence? And that's the first thing that I'll discuss. If we decide that there is genetic influence, Or let's say there's familial occurrence, that this occurs more commonly in family members than one might expect. What is the reason for this? And not all coexistence of diseases within families is genetic. Members of a family share other things besides their genes. They share their environment. They live together and would be exposed to environmental influences more. And as you'll hear, when there are multiple genes that contribute to why someone gets a disease, and you've got to have gene X and gene Y and gene Z, because they're all independently inherited, and unless you get certain combinations of these, you don't get the disease, and maybe you'd need an environmental trigger, it may not be entirely obvious that there may be genetic influence, as most people would probably say that they don't have any affected relatives. It doesn't mean that genes are not involved, and understanding this interaction between different genes and between genes and environmental factors is a key to understanding the answer to the question. And then if one establishes that there is genetic influence and the type of way that that genetic influence is it transmitted directly from generations to generation, or is it a so-called complex genetic disease, which we'll talk about in a minute? How does one go about identifying the causative genes? Because before we can come up with a genetic test that we'd apply to you to predict whether your relative is going to develop the disease, we have to identify those causative genes in the first place. As it turns out, it's a fairly trivial thing these days, once one's identified a gene, to devise a genetic test. That is pretty easy. The difficult thing is to come up with the causative genes and understand exactly what it means. How strong of an effect is it when you test for gene X? How well does it predict it? And does that gene occur exclusively in people with the disease, or does that gene also occur in people who do not have the disease? And to discuss all of these issues, I'm going to talk from my knowledge of two diseases in which both of which I've worked in terms of genetics. One is multiple sclerosis, for which there has been considerable work done and millions of dollars invested, probably tens of millions. And I'm going to talk about neuromyelitis optica, where there are relatively few investigators working on genetic aspects, and much less money invested, and I suppose we're less far. But I think those are two topics of interest to members of this audience, so I will try to present answers to these questions for both multiple sclerosis and neuromyelitis optica. And the first issue was, is there indication for a genetic influence in autoimmune disease? And the simple answer is, yes, there is. But like I said, this is not intuitive. Sandy Siegel said this isn't intuitive to me. Most of the people that call me don't report any relatives. And that's why you have to go beyond just your exposure to people that you know, and you really need to do this in a systematic way. And as Sandy told me, you know, if people tell me that there are symptoms, I'm not always sure that it's related, that they in fact had transverse myelitis, even if they tell me that a relative has transverse myelitis. So it really means tracking down medical records of relatives. And I think I'll show you an example of how this was very informative to me in one patient that I saw shortly. And when one's asking, is there an increased frequency of disease in relatives, you have to say compared to what? And generally speaking, we're talking about compared to the general population. But other things that can be informative is asking whether close relatives, brothers and sisters, have a higher frequency than, say, a more distant relatives, aunts and uncles, than even more distant relatives. And the rate of decrease of that frequency can give some indication about the type of genetic inheritance. And as it turns out, one group of individuals, and it's very hard to find these because there's only about 1 in 80 people that has a twin, but twins turn out to be very informative because identical twins share all their genes, whereas dizygotic twins or fraternal twins, non-identical twins, share only half their genes. And comparing those two groups, finding those rare individuals who have a twin, can be very informative. And then I've raised this question before. If you find an excess, is it due to something inherited or shared environment? And then if it is genetic, is it due to something individual within that family that may have a strong effect that goes generation to generation, so you see multiple generations affected, or is this a weak gene that somewhat increases predisposition? We see this weak gene in many people that may not get the disease, but overall there's a slight increase or a definite increase in frequency of that gene, but that gene has only a mild predisposing effect. And it's a very different approach in terms of counseling. So let's look at the answers for multiple sclerosis to those questions. The frequency in relatives, let's say brothers or sisters or children, is less than 5%. Said other ways, 95% of relatives of MS patients are never going to develop this disease. But as I said, it's compared to what? And if you think of what the frequency of this disease is in the general population, which is about 1 to 2 in 1,000, or 0.1 to 0.2%, a 5% frequency is 50 times greater. So in general, we say that although the risk is low to relatives, it's about 20 to 40 times, depending on the exact relationship, compared to the general population. Not enough that an individual person would worry about, because the absolute frequency is less than 5%, but if you're a scientist studying genetics, you'd say, well, if it increases their risk 20 to 40-fold, I want to understand why that is. That's going to tell me something about multiple sclerosis. As it turns out, the risk to an identical twin is about 1/3. If you have MS, if you have an identical twin, most often those identical twins are not going to develop MS, 70% of the time, but about 30% of the time, which is a pretty high percentage, they will. That compares to dizygotic twins or non-identical twins. Remember, they share only half their genes, and the frequency in that group, if one member of the pair gets MS, the other member of the pair has about a 5% lifetime risk. So a 6-fold difference. And I'll tell you that that tells us in advance, because if it was a single gene, it should be 30 to 15%, because the difference between half and all your genes is about 2-fold. The fact that it's a 6-fold difference tells us automatically there's got to be more than just one gene, either multiple genes or gene environmental effect. And the risk declines with distance of relationship. And this review by Coles and Compton that was published in 2002 in The Lancet shows that the risk to an identical twin is about 30%. Here are some rare situations where you have a sibling plus 2 affected parents. The risk is 25%, sibling and 1 identical parent, about 15%, and by the time you get to a dizygotic twin, it's down to 5%. Now you can see the risk decreases from there, not to 0, because we know even somebody in the general population has about a 1 in 1,000 risk of getting multiple sclerosis. And this drop-off of a curve is typical for what we see with a multiple genetic predisposition disease, or a so-called complex genetic disease. Now, is the effect due to genetics or environment? And in fact, it's being studied in people that have brothers or sisters that were adopted into another family, often at birth, and there seems to be excess risk even though they were adopted at birth. And this is fantastic work tracing all of these unusual situations by a consortium of investigators from Canada, led by Dr. George Ebers, who I had the opportunity to work with while I was in Canada and trained under him. And furthermore, individuals adopted into those families seem to have no increase in risk. And again, it takes many, many individuals to be studied to come up with this conclusion that there is no increased risk. But this seems to speak to the fact that the excess in families is primarily genetic and not due to environmental sharing. We know that there is predisposition primarily to Northern European populations, but there are several pieces of evidence that also point to the fact that this genetic risk can be changed by environmental factors. In particular, if you migrate from a high-risk area like England, to a low-risk area, South Africa, which was the original study, you can change your risk. And there have been studies of migration from Asia to Hawaii, from India to the UK, many examples, all of which seem to consistently come up with the same answer. We could talk about the problems with all of these studies, and there are always problems, but I think it's a pretty consistent answer. And we don't understand the exact environmental influence, and Ben will be talking about this, but he may talk about certain viruses and vitamin D levels as being established to be important. Well, what about neuromyelitis optica? There had been reported familial cases of neuromyelitis optica, and until recently, it had been felt that neuromyelitis optica was primarily a disease that occurred in individuals of Asian and African background and perhaps some excess in Hispanic. I don't believe this is true. I think it's simply the fact that neuromyelitis optica was not recognized. The best evidence now is that although the proportion of MS-like illness in Asia that's neuromyelitis optica is much higher, probably about 20% of MS-like illness, where it's only about 1% in North America, the absolute number, if you look at the number of people in the population that get neuromyelitis optica, seems similar and is about 1 in 100,000. So I don't believe the previous bias that there was an excess in Asian and African is reliable. However, there had been 4 reports until recently of familial neuromyelitis optica, mainly in the pre-NMO-IgG era, dating back to McAlpine, who's a famous historical figure in MS, and more recently, Dan Michael, who was at the University of Michigan at that time, who reported a mother and daughter. This was the first one where there was NMO-IgG testing involved, and one of the family members who was testable was positive. The other one was deceased. My ground zero case was this lady who I saw, a 29-year-old woman who in August 2006 had a severe right optic neuritis and had visual loss, and her neurologist in Iowa ordered NMO-IgG, and it was positive. And she has since developed recurrent optic neuritis and myelitis, and we're pretty confident about the diagnosis of neuromyelitis optica. But when I saw her after that first optic neuritis, she told me about a sister who was in fact also seen at the Mayo Clinic in 1991 by a colleague of mine who's an excellent neurologist. And I think it just shows how much things have changed. She was diagnosed with MS and apparently died of MS. And I thought, boy, that's an unusual thing to die of MS as a young person. But I said, could we look at her records? And we got permission to look at her medical records. And that is her sister, the sister of this index case, had had episodes of optic neuritis and myelitis with long lesions, and died in 2000. And in fact, she had an antibody test, not for NMO-IgG, which we didn't even know at the year that she died in 2000, but she was tested for paraneoplastic antibodies, which Dr. Vanda Lennon runs that lab, and she was the one who discovered NMO-IgG. And we were able to de-archive her serum sample and test it, and it was positive. So clear evidence that both sisters were positive. And that was the patient that perhaps got me most interested. And this paper just appeared a couple of months ago in a journal, Neurology, from our group. And this is a collaborative effort with investigators from the United States, South America, Korea. And we reported 12 cases where there were 2 individuals affected with neuromyelitis optica, and actually one family had 3 individuals. 21 or 84% were women. They could occur in one generation, the brother-sister pairs, or 2 generations, parents-child, but we did not see any over 3 generations. We had sibling pairs, parent-child pairs, or avuncular, which would be aunt-niece pairs, or uncle, well, we didn't have any uncle ones, so so-called avuncular pairs. We saw And this allows us to explore transmission because, say, one form of transmission can occur on mitochondrial DNA, in which case we would always see that the transmission comes through the maternal line. But we saw instances where there was apparent paternal transmission. So that told us that mitochondrial inheritance shouldn't explain this. And as best as we could tell, these cases were indistinguishable from otherwise sporadic NMO without relatives. We don't know what the environmental events are that may be important or whether environmental events are important. So one of the questions you can ask is, do they get NMO in the same calendar year, which would suggest some kind of common exposure, or are they more likely to get it at the same age, which would be more like genetic programming? So this is another way that one can inquire or look into the relative importance of genetic versus environmental effects. And to make a long story short, there was no clear answer here. The only thing we saw is that there was a tendency to get this disease in late middle age, which we knew anyway. That's true of sporadic cases as well. Most of these patients were positive for NMO-IgG, and based on looking just at individuals who came to us because we diagnosed NMO, and then we asked them, do you have an affected relative? We did not take any who were referred to us specifically because they had a relative with NMO. There were very few like that. We calculated that at minimum, these cases represent 3.1% of NMO patients. And it varied from 1.3 to 6% at the 4 different participating centers that contributed families to this study. What is the mode of transmission? And as I mentioned, the key thing is this a disease that's genetically determined, that tracks generation by generation with a very strong predisposing gene, like the Huntington's gene that causes Huntington's disease? That's one that if we test for a gene and we find it's positive, we can pretty much guarantee that if this person doesn't die of a car accident or something, they're going to eventually get Huntington's disease. It's genetically determined. Or is it just a gene that predisposes, makes it more likely, changes your immune system in some way that you're more prone when you get exposed to an environmental factor to get the disease? So that's a very key question. And we decided that it's more likely the latter. And that's because most individuals do not have affected relatives, that it does not tend to track over multiple generations. This means the penetrance, or the ability to cause the disease, of the gene is low. There are no apparent differences. For some conditions where there are sporadic and familial forms, the familial forms look sort of different. And if you're a smart clinician, you eventually get to say, well, I can recognize those ones that are genetic because they have these or those characteristics. But to date, we've not been able to find any characteristics that are distinctive for the familial cases. And it's rare to see pedigrees greater than 2 generations. And in situations like this, where complex inheritance is most likely and most relatives do not get the disease, in general, empiric prediction, like the statistics that I gave you for MS, 2 to 4% of relatives affected, your risk is about 2 to 4%, is usually much more helpful than any form of genetic testing, because it usually means there's multiple genes, all of weak effect, and that's certainly turning out to be the case in MS, as we'll talk about in a minute. And testing for genetic risk to predict recurrence is not very effective. The most helpful thing are these collecting large series, figuring out what is the percentage risk, like I presented for MS. And I think that's one of the major take-home messages, is the statistics that I've presented to you are going to be the single most helpful answer that I'll be able to give you when you ask me that question, what's the risk to my relatives? I'll say, in this big experience of testing thousands of or hundreds of people we've seen with NMO, this percentage have an affected family member. And we're probably unlikely to be able to refine it, certainly for MS, where there's been a lot of study, much better with genetic testing. Well, we don't just give up, and what is the strategy to identify genes contributing to the disease? There are many kinds of genetic variants. The single most common are single nucleotide substitutions. Now, DNA, which is our genetic material that codes for everything, is composed of 4 different nucleotides. And I won't tell you the names, but they go by the initials ATGC. And here, G is switched to an A. That would be called a purine transition. And that kind of substitution could be a mutation that arises or is inherited that could do nothing or could cause disease. Other ones are small either insertions or removals, deletions of bases. And another one is insertion of repeat sequences where a gene becomes duplicated. We're now talking about copy number variations. You might even see that now on CNN or Discovery Channel or something where a gene is duplicated, sometimes several times. So many different kinds of polymorphisms, SNPs or the single nucleotide polymorphisms being the most common. Here's a sequence of DNA comparing 2 individuals, and you can see they're all identical except at this position. This individual has a T and a T, this one has a C and a C, and this one has one of each, a T and a C. And because we have 2 copies of DNA, we have two bases at each point. And in some situations, if this were pathogenic, having 2 can cause disease. In some situations where maybe there's some toxic effect of having even 1 C copy, both of these situations are one where disease could occur. Now, these are incredibly common. 1 in 1,000 of our DNA bases is a variant base, and they may have absolutely no function, or they may change the amount of a gene product that's produced by, if they're in the promoter region, or they may change the structure of a gene product, and they can do so either in a radical way to make the protein not work at all, or they can do so in a gentle way that just sort of changes it a little bit. And you could imagine this might make a difference as to whether you get disease from this or you don't. And we've talked about this. And the important thing is when we find things, we have to be very careful because we know many of these are apparently silent and not associated with any change at all. But research has told us that a number of different diseases, here's thyroid disease, here's a tendency to form clots, can be associated with very specific SNPs that we know are associated with that disease, because when the studies have shown that the frequency of this SNP is very, very much higher in a group of people that have the disease. And some of these SNPs are very low frequency. Here's 2%. Here's a SNP that is fairly common, present in maybe 60% of individuals. So, and a general rule is that common diseases generally have common genes associated with them, because if you had a gene that was causing a common disease, if it was that common and causing a very serious or fatal disease, the human race wouldn't be around. But rare diseases tend to have rare genes, and MS is a common gene. And we can then design tests, and here is a test that shows the length. This gene, we were actually studying, this is work that we did studying MS with CCR5, but one variant that's fairly common in the population, Delta 32, as it turns out, completely protects one from getting HIV. This is a chemokine that's involved in the immune system, but it turns out that this mutation is the receptor that's used by the HIV virus to get in, and those individuals with the shorter product that runs down longer would be resistant to getting AIDS. So it's easy to set up a test once you've established the significance of this, and now these kind of gels, although 10 years ago that was the standard way these were done in clinical labs, now we have all kinds of automated fluorescent ways to detect genes. But it just shows each one of these lanes is a single patient. You can screen very large numbers of patients quite quickly when you know what you're looking for. But we got to find it first, and I'm not going to spend a lot of time, I think, for this audience talking about it, but we can adopt a different approach where we have a large family, like with Huntington's disease, where it segregates over many generations, where we can use very powerful techniques called linkage, which have a very high chance with a single family, or just a couple of families, of pinpointing where the gene is. When you're dealing with relatively common diseases like multiple sclerosis, typically we do association studies, and we get one of those SNPs, and we know where most of the SNPs are because these are all cataloged and available online through NCBI, which is a national governmental database, and we can test for many genetic markers simultaneously. Actually, there are chips now that do, say, 300,000 genetic markers in one large experiment. And you might find one individual in the affected and one individual in the unaffected populations that you test. With this marker, you'd say, well, that gene doesn't look very promising, but here's this other genetic marker. It's present in all these individuals in the affected population, in only one in the unaffected. It is present in one of the unaffected. Well, that one looks promising. Now, do you study individual genes that you think are likely to be important, or do you just survey in an unbiased way across the entire genome, which is huge, 3 billion base pairs? And there are advantages and disadvantages for both. Certainly cost-wise, the whole genome analysis is very, very costly, although decreasing in cost. So let's talk about what we've learned with MS. There's one important risk allele, and I'm probably going to get in trouble for saying that. I'm not suggesting the other work that's been done is unimportant, but in terms of how strong an effect it is, something we've known since the 70s, HLA, which is a gene that controls how the immune system responds, is the most highly associated genetic marker. It's 3 times more common in MS patients compared to the general population, but it's present in 15% of the normal healthy population. And therefore, it turns out not to be very helpful to test to predict risk, because if you're positive, the chances are low that you'll develop MS, greater than 99% chance you won't develop MS, even though it's 3 times more common in MS patients than the general population. It's not even helpful to rule out MS because 50% of MS patients don't have it. But this is the strongest one we know. That we got cheap in the 70s. We've spent tens and probably hundreds of millions of dollars to get all of this recent stuff that's come up from the whole genome association study. The most important thing it's told us is most of these markers that are coming up tend to be in genes that control immune system. And that wasn't that obvious because it was thought, well, maybe it'll be some brain protein or some other kinds of proteins, but they have, and I suppose it shouldn't be a shock because we've thought the immune system was important for many years, but all of the predisposing genes have tended to be immune genes. But look at their effect. They increase the risk by maybe 20%. They're 20% more common than they are in the average guy. So you got an almost 50-50 chance if you pick up this gene that you could be MS or you could be not MS. So they're not at all helpful individually for testing. Now, maybe somebody very smart will figure out how to put these together and figure out certain key combinations that may substantially increase the risk. And there's already evidence that if you look at evidence of Epstein-Barr virus infection plus this gene, the HLA gene, which is the strongest one, the combination may increase your risk tenfold. And maybe that's getting a little bit better toward prediction. But I think the important message was that that the immune system is important in MS, which it's a long and expensive way to figure this out, but probably worthwhile, and certainly worthwhile. So what about NMO? We've invested much less into it. MHC, or the HLA system, the same one that we talked about with MS, seemed an important one to look at because it's important with many different autoimmune diseases. And aquaporin 4, which is the target antigen of NMO, seemed important. And there have been multiple studies that I've summarized here, and I'll give you the details. In Japan, although there's an association of so-called Western MS with this MS susceptibility gene, there is no association of NMO. And similarly, in a French study, it was not associated with that gene. It was associated with a different HLA gene, DR3, a threefold increased risk compared to controls. And in fact, study by Bruce Cree was a small study in African Americans, but they were tested for NMO-IgG. And one of the things that he had pointed out is, I didn't find a single one that carried this MS susceptibility gene in the group of patients that were NMO-IgG positive. We've recently published this much larger study on about 200 patients with NMO, and more or less have confirmed the same thing, that in MS patients, threefold more common, this HLA DRB1 1501, than controls. But in NMO, we actually found a statistically significant decrease compared to controls in this same gene, providing further information or further proof that these are distinct diseases, which we've kind of known anyway. But we've also undertaken comprehensive sequencing, and we've come up with some interesting observations that we still don't exactly know the significance of. In particular, we found 3 individuals with mutations at this point in this arginine codon. Groups of 3 of these building blocks for DNA code for a specific protein, and AGA codes for arginine. In the aquaporin 4 gene, we found ATA in 2 individuals in one family that both have NMO. And we also found another individual who had a different mutation here from AGA to ACA, but at exactly the same site. And as it turns out, these cases were African American, and this patient was Jewish, and they were not related. And in fact, they had different mutations, but at exactly the same site. And we said, well, does this occur in healthy individuals? And again, we came up with a way that we can look at large numbers of patients. In fact, we looked at 1,363 controls. And this enzyme cuts only where the wild type sequence is present and doesn't cut where there's a mutation. So here's one of our patients with the mutation, but you can see all these controls that don't cut. And basically, we found 3 NMO patients positive, none of the controls. And in that group where there were 2 affected individuals, this index case and her aunt, both of them carried the gene, as did the father, who we figured, it had to come from his side of the family because the aunt was a paternal aunt. So we said, well, how did she get the gene? The father would have had to be a carrier, and as we predicted, he indeed was. And as it turns out, this individual, her sister, also carries this gene, but has no symptoms. Now, is this gene important? We don't know. It does seem to segregate in the family, and we've done quite a lot of work, and we continue to do work to try to figure out how this gene is producing the disease. It seems convincing. It's obviously something present in a very small number of cases, only 3 out of 200 patients with NMO that we've tested. When we first discovered it, we thought, well, maybe we'll find something that explains a lot of NMO, but unfortunately it does not. But it may yet give us some clues as to how aquaporin 4 gets processed in some aberrant way so as the immune system responds to it. But we don't have a definitive answer on that. One of the ways that we can look to see how important this is it a residue that's conserved throughout evolution? We look at various species, and as it turns out, this is a conserved residue, which is one clue that it might be quite important. If it's not conserved, it's probably more likely to be silent, but we don't have an answer. So, By way of conclusion, I would suggest to you that both multiple sclerosis and neuromyelitis optica are complex diseases. They're not going to affect many relatives, but there is a slight excess of individuals amongst family members. If we take all NMO patients and all their family members, we're going to find a very slight excess numerically compared to what we would expect in the general population. In multiple sclerosis, the main predisposing gene is HLA or MHC. They're the same thing. They're genes that control how the immune system responds. But there are many other immune genes that contribute a weak effect. And at the present time, they're not helpful in testing or predicting susceptibility. In NMO, we actually find the opposite association, a negative association, or it's less common to have this MS-associated gene compared to controls. And there does seem to be, according to a French study, an excess of a HLA type called DR3. But in NMO, there may be some rare mutations that contribute to NMO risk in a very small number of families. Thank you very much for your attention. I know this was kind of tough going to talk about genetics, and I'd be happy to try to answer any questions. It's hard for me to see with the lights, Ben, but. Mm-hmm. Yeah. Right. So the question is, are there any indicators, not just for MS or NMO, but autoimmune conditions in general? And there's been a lot of interest in shared genetic markers between, say, multiple sclerosis and other diseases. And it's interesting that some of the genes that came up in that long list for MS are also genes that seem to be important for, say, type 1 diabetes, which we think is autoimmune, and other diseases. But in some cases, the same genetic marker that seems to be protective, predisposing for MS seems to have the opposite association for diabetes. Even though it's the same gene, it seems to have kind of the opposite effect in terms of susceptibility. But there are families where multiple autoimmune diseases occur in certain conditions where that happens. Neuromyelitis optica, we see that about 25% of patients will have a personal history of other autoimmune diseases, and we've seen NMO patients with 3 or 4 diseases, lupus and myasthenia gravis, and then neuromyelitis optica, and we don't think that's a coincidence. So I think there are likely common autoimmunity genes that control the overall volume of autoimmunity and how the immune system responds, makes the immune system hyperreactive. And there are probably other ones that control and determine to some extent what specific autoimmune syndrome one's going to get. Is it NMO or myasthenia gravis? And probably a combination of those are going to be important. Holly Schmidt, let me specifically thank all of the people. I had it on my first slide, but I didn't spend enough time. Holly's ACP organization has been very helpful and generous in providing us with samples for this study, as have other collaborators, and I thank them very much. So the question was about transverse myelitis. I think the problem with transverse myelitis is that it's a syndrome. I don't know what the disease transverse myelitis is. When someone tells me they had transverse myelitis, I can't put it in a disease. It's like a symptom, as it were, or a syndrome. We used to call idiopathic transverse myelitis. We now recognize that 30% of them with long spinal cord lesions are neuromyelitis optica, it seems, or in our experience have tested positive. So I think it's a hard thing to study. Some of them may be stroke-like syndromes. Many of them are going to be MS, and because I think it's so diverse, I think it's not attractive to study in terms of risk factors. I think what we have to do is try to break up transverse myelitis, not the organization, that we want to keep, but we have to break up this disease and understand its component parts, because it's important in predicting what's going to happen to a patient. You know, if they tell me I had transverse myelitis, I have no idea what they can expect or anything, but if I look at their scan, I appreciate the size of their lesion, I can say this one, I'm kind of worried about MS for the future. This one, I have no concern about MS, but maybe NMO. And to the extent that we're going to be able to refine that, I think we're going to do better. The other question was about MS. ADEM. That's another one that I would say, of all the cases that are referred to me as ADEM, I would say a small fraction of them leave with that diagnosis. I think many of them are fulminant MS. I think there is such a thing as ADEM, but it's true ADEM, but it's very rare. And the term, I don't know what was said earlier today in this session. I hope I'm not contradicting anybody else. But here I am giving my answer. Most of the cases with it, I think, may have something else.